So L10b will be overview of eukaryotic transcription of class II genes, pre-mRNA processing, and mRNA expression. So in eukaryotes, when we transcribe the DNA of class II genes, we have regions that are called exons and regions that are called introns, and then we have sequences before the introns and exons and sequences after the introns and exons, and all of that is transcribed. And we'll find that exons are the sequences of the transcript that are used for coding sequences, and the introns are intervening sequences, and they're going to be eliminated and the exons will be spliced together. So this whole thing is transcribed as a transcription unit. The transcript is called pre-mRNA. And then once we make the pre-mRNA, we go through some processing steps. We add to the 5' end, we have what's called a cap, and we do our RNA splicing where we splice out the introns and splice together the exons, and then we add what's called poly-A's to the 3' end of the mRNA, and once we do a successful processing, we export it through the nuclear pores, and that gives us mRNA for translation or protein synthesis. So RNA polymerase II and the C-terminal domain comprise an mRNA factory. So this is RNA polymerase, and we've talked earlier about the C-terminal domain, which can be phosphorylated, and one of the factors that phosphorylates it is transcription factor H. So upon phosphorylation, this recruits various proteins. So we have a protein complex that has to do with capping. We have a protein complex that's related to splicing. We have a protein complex that's related to adding the poly-A's onto the pre-mRNA. So these factors are actually associated with the RNA polymerase, and many of these processes take place as we're transcribing. So this shows an example of we're transcribing the transcript. Here's a transcript. This would be the 5' end, and the capping factor is now associated with the nascent RNA, and it's capped. So these processes happen while transcription is taking place. RNA polymerase II, adding of the cap to pre-mRNA. So here's our 5' end. This is the 3' end that's being extended. This is the cross-sectional view of our RNA polymerase II. Here's our cap synthesizing complex. And as the RNA is being extruded from RNA polymerase II, the cap synthesizing complex binds to the RNA, adds the cap, and then we have the cap binding complex, which is a complex of proteins that binds to the cap and associates the capped end of the pre-mRNA with the C-terminal domain. So notice that this is a loop that's growing as this part here is being synthesized and this is extruding. So the mRNA cap, what's it all about? It stabilizes the 5' end of the pre-mRNA, and it provides a ribosomal recognition. So here's our eukaryotic mRNA. Here's the cap. So the cap, it has a methylated G on the end, and it's linked via a triphosphate linkage to the 5' end of the pre-mRNA. So here's our 7-methylguanosine. There's the 7-methyl. Here's our 3 phosphates. There's the 5' end of the pre-RNA, and now the cap is added, and what this cap does is it doesn't give the 5' end a 5' end. This is the 5' end, and because of this linkage, which is a 5' to 5' triphosphate linkage, we don't have a 5' end for exonucleases to degrade the 5' end of the molecule. So this stabilizes the molecule. And in addition, we'll have our cap binding complex that again helps stabilizes the 5' end and is also involved in ribosomal recognition. So we've added the cap. We're going to do splicing next to give us our continuous splicing sequence. And then we'll have a non-coding sequence here, and then we'll add the poly-A tail. RNA polymerase II, splicing during transcription. So here's the cap binding complex, C-terminal domain. This is called the spliceosome. We'll talk more about them in a minute. They're RNA protein complexes that are going to splice out this yellow part, which is an intron. So the green parts are exons. So as the loop is being formed, here's our spliceosome complex, and notice it's linked this end of an exon, which is the 3' end of the exon, to a 5' end of another exon, and the spliced intron will be degraded. Exon splicing continued. So here's our primary transcript, pre-mRNA. We have a particular set of sequences at exon-intron junctions. So this would be an exon part, intron part, intron part, exon part. Those are called exon-intron junctions. And within our intron, we're going to have a particular loop, and the loop is going to have the sequence of CAA. Case the first A has a free OH, and that free OH will attack this phosphate between this U and this particular G, and when it does that, it's going to make a loop, so it breaks the bond between the U and the G. The G becomes attached to this OH, and that gives us a loop. And once we make that loop, we have this 3' OH group that's free. It can attack the phosphate of this U. And now we made a covalent bond between this exon and that exon. And this loop, which is also called a lariat, it leaves. So here's the part that leaves. And this particular bond between, this is a 2' OH, and this is a 5' on the G. That's a 2' to 5' phosphodiester bond. The adenosine in the lariat has 3 phosphodiester bonds, so it has a phosphodiester bond here, a phosphodiester bond here, a phosphodiester bond here. So this bonding is really unusual. So here's our intermediate with the loop. The loop is removed, and there's our lariat, the 3' OH of the 5' exon. So this is the 5' exon, acts as a nucleophile, completing the reaction. And now we have a spliced RNA, and we have the free lariat loop structure, and this will be degraded by nucleases. This shows it in a little more detail, and actually there's several different kinds of introns, and this intron is slightly different from the one we've seen. So the sequences we see at the intron-exon junctions are a little bit different, but the mechanism is the same. So this is exon splicing. The role of small nuclear ribonucleic particles are called snRNPs or SNRPs. So these are RNA protein complexes that bind at the exon-intron junctions and bind at the A with the OH group that's going to attack over here, and they actually catalyze the splicing mechanism. So one of these RNPs is called U1. The other RNP that binds at the A, that's going to be our nucleophile, is called U2, and we take ATP and we assemble this complex. So it takes energy to assemble these ribonuclear particles into this complex. And then we take ATP again, and we bring this A closer to this U. So here's the A coming closer to the U. The A will attack this G. It's made the loop, and now we have the U OH group on it, and it will attack right there, whatever nucleotide this is. So this is showing what's on the intron. It's not showing the nucleotides on the exon, so this OH will attack right here. And so here's our loop. It has the UG on it. There's the UG. It has the AG. There's the AG. There's the intron release. Here's the lariat. And now it doesn't show it, but the 5' and the 3' are connected here at this gap. So each one of these U1s, U2s, all these different U's are nuclear ribonucleic particles, snRNP, and they have catalytic activities. So this is a case where RNA acts as an enzyme. So this is quite interesting. So what we showed in the previous slide, we didn't show that these are actually RNA protein complexes that are involved in this particular reaction. So those are called small nuclear ribonuclear particles. The RNAs are called small nuclear RNAs. Formation of poly A tails and polymerase termination. So here's the RNA polymerase. As it's transcribing the DNA, it encounters sequences that are related to cleavage and polyadenylation of the pre-mRNA. Here's our C-terminal domain. It has protein factors. This is called cleavage and poly-A specificity factor, and it's involved in the recognition of the sequence AUAAA, and that's a sequence for polyadenylation and cleavage. We're going to add additional cleavage factors. RNA will be cleaved. Poly-A polymerase is going to bind to this complex. We have additional cleavage factors. Here's the poly-A polymerase. Here's the poly-A binding proteins. And now, if we cleave just about right there, and we add poly-A, here's our sequence AUAA, AUAA. Here's the cleavage and poly-A factor. Here's our poly-A's. That was added by poly-A polymerase. And then once we make the poly-A's, we have poly-A binding proteins that bind to the poly-A. So after we make this poly-A on the 3' end, it's bound to proteins. It's not free in the nucleoplasm or the cytoplasm when it goes through the nuclear pore. And this stabilizes the 3' end of the RNA molecule. So now, when we have the poly-A's, we made a mature 3' end of an mRNA molecule. So we start with recognition proteins. We recognize the sequence AUAAA. They bind to the forming pre-RNA. They cleave the molecule. Remember, this is the 3' end. This is the 5' end. They cleave the 3' end. RNA poly-A polymerase binds. It adds the poly-A tail. As we're adding the poly-A tail, we have poly-A binding proteins that are going to bind and stabilize the poly-A's. So again, we use the C-terminal domain as our processing factory. We used it for adding the cap, we used it for removing introns, and we've used it to add the poly-A tail. Structure of ribonuclear protein complexes determine nuclear export. So often on the board, when we talk about mRNA, we just draw a line and say this line is the mRNA and ribosomes bind, and then we get translation to protein. Well, that's not true. These mRNAs are riboprotein complexes. So they're complexes of proteins and mRNA. We've already seen that. Here's our poly-A binding proteins. These are proteins that have to do with the splicing phenomenon and export. So we have RNA export factors, we have splicing complexes, and we have the cap binding protein. And all of these factors that are associated with the mRNA indicate that the mRNA processing happened correctly. It doesn't mean that there's not sequence mistakes in here from mutations, etc., but it means that the processing itself was done correctly. And that allows this mRNA protein complex to leave through the nuclear pores. So these RNA export factors, they actually have to do with recognition at the nuclear pore. We have what are called nuclear restricted proteins. They stay inside the nucleus and they will bind to other processed mRNAs. And here's our poly-A's, our poly-A binding protein. Here's the poly-A. Here's the cap binding complex. Here's the cap. Here's our other factors that are bound with the mRNA. And once it enters the cytosol, we have other proteins that associate with it. So here's the cap binding proteins. This EIF4A, EIF4G, these are elongation and initiation factors that have to do with ribosomal function and recognition. So the cap binds to these proteins. And notice what we've made is now the mRNA is not a long squiggly line that we're going to translate. It's made a loop. This is what we're going to translate with ribosomes. We translate this loop with ribosomes. We don't translate a linear mRNA. Alternate splicing to form distinct proteins. So it's not unusual that our DNA sequence has multiple splice sites. So here's the poly-A site. Here's a 3' site here, there's a 3' site here, and this whole sequence is actually a coding sequence. It's not an intervening sequence. This is an intervening sequence. These are coding sequences, but we can splice between here and there, or we can splice between this arrow and that arrow. And when we do, we will get an mRNA with a different sequence, and the mRNA can make a protein molecule that's different size and different sequence, and that's what this particular figure shows. So here's our primary transcript, our pre-mRNA. It has a cap. We're going to add the poly A tail. Now we do our splicing. We can splice out this part right here, and that gives us this longer sequence. We can splice between here and there, and that will give us this shorter mRNA. So this is a way of making different proteins or slightly different sequences of proteins from one gene. In the case of some genes, as many as 10 different proteins, depending on how we do our splicing when we remove introns. So that concludes L10b.